Okay, so I think we'll get started. I know we've got a lot of content to get through today. So hello and welcome to everybody who's joining us today. Thank you so much. This is the second webinar now that we're doing under our carbon monitoring training series here at WWF. So today we are going to be digging in and looking at soils and sediments. So we'll be looking at both both terrestrial and marine environments. So for those of you that don't know me, my name is Emily Giles, and I am the lead specialist on our science, knowledge, and innovation team. I'm based in Perth, Ontario, on the traditional lands of the Anishinaabe Algonquin. So welcome, and thanks again to all of you for joining. Today's session will be recorded, and we will share the recording with participants afterwards, and we're also going to upload it to our learning library. So hopefully many of you joined our first session and learned about our learning library there, but we'll post the link again here in the chat for the learning library where you can access the tools and resources that we've developed as part of our carbon monitoring program. If you have questions as we go through the webinar today, please do add them to the Q&A function that we've got here as part of Zoom. We'll try our best to answer them as we go through the webinar, but if we don't get to them in the chat function itself or in the Q&A function itself, we'll answer them live at the end of today's webinar. We've got some time at the end to go through those questions. Also, if you speak French, we've got Catherine Paquette here with us today, who is going to help us with French and English translation. So no problem at all if your question is in French. And any technical issues, please send us an email at science@wwf-canada.org, and we'll try our best to respond to it as quickly as we can. So in terms of what we're going to cover today for this second webinar, first of all, we'll just go over our objectives and what we hope all of you will get out of today's session. We'll do a quick round of introductions so you know everybody on our team here, and you'll know as well who's joining the workshop today. We'll do a quick overview of the workshop, workshop 1, which gave a bit of a Carbon 101 overview. So we'll go over that quickly. And then our session today is divided into 3 parts. So in part 1, we'll do a background, again, looking at soil carbon and why it's important to measure it. And then part 2, we'll get into how you actually go out and measure that soil carbon. So we'll go over sampling designs, what equipment you're going to need, and the methods for that as well. And then in part 3, we'll look at how do you interpret and report on those results, which is probably one of the most common questions we get from folks who are doing this kind of work. And then at the end, we will have that Q&A session, as I mentioned. So in today's webinar, we're hoping that you're going to learn what soil carbon is and why it's important, as well as how to actually go out and measure that soil carbon for the purpose of determining the soil carbon stock. So we'll give you some very practical guidance on how you can do that. We'll also, you'll learn today about the different types of soils and sediments, as well as the different tools and methods that are required for measuring those different soil types and conditions. And then we'll go over some practicality in terms of how you actually go about designing a soil sampling project. And then again, finally, you'll learn how to interpret and report on the results of that project. So on that note, I'm going to pass it off here to our team to start introductions. And while our team is doing introductions, we'd also love to hear from those of you on the line and hear who you are, what your affiliation or role is, and where you're located across the country. So please do add them to the chat. And I'll pass it over to Claire from our team first to start the introduction.
Thanks, Emily. Hi, everyone. I'm Claire Wark, a specialist on WWF Canada's Science, Knowledge, and Innovation team. I'm based in Collingwood, Ontario, along the south shore of Georgian Bay, which falls within the territory of the Anishinaabe Nation, along with the people of the Three Fires, the Odawa, and the Potawatomi nations. And this land has traditionally been managed by the collective Saugeen Ojibwe Nation. I spend most of my time here at WWF Canada connecting with communities, leading carbon training and fieldwork initiatives, and then also supporting our carbon community of practice calls. And I will pass it over to Cal.
Hi everyone, my name is Cal or Cahal, which is spelt a little weird, so I usually go by Cal. But I'm the Associate Specialist at Science, Knowledge, and Innovation at WWF-Canada. My work stems from my experience working on ecosystem carbon throughout Canada, providing tools and information to build capacity of individuals who are also interested in this kind of work. Yeah, I'm excited to be here today to discuss in a little bit of detail on soil carbon and all that. So I'll pass it on to Catherine.
Awesome. Thanks, Cal. So hi, everyone. I'm Catherine Paquette. I'm a specialist of freshwater ecosystems on the restoration team here at WWF Canada. And today I'm joining this webinar sort of as a translator. So if anybody has any questions that they would like to ask or write into the Q&A in French, it'll be my pleasure to translate them for the group. And then vice versa, if there's anything that isn't you're not getting, I'm happy to translate it back into French. Donc, bonjour tout le monde, mon nom c'est Catherine Paquette. Je suis spécialiste des écosystèmes d'eau douce sur l'équipe de restauration ici avec WWF Canada. Moi, je joins ce webinaire aujourd'hui comme traductrice. Donc, si vous avez des questions que vous voulez poser en français, s'il vous plaît, n'hésitez pas. Il me fera plaisir de traduire vos questions pour que le reste du groupe puisse les comprendre et vice-versa. Donc, s'il y a quelque chose qui est dit durant le webinaire que vous ne comprenez pas ou que vous vouliez que je traduise, s'il vous plaît, laissez-moi savoir. Vous pouvez m'envoyer un message dans le chat, puis ça me fera plaisir de le traduire. So over to Marianne.
Thanks, Catherine. Morning, everyone. My name is Marianne Fish. I'm a senior specialist for coastal ecosystems on our Science, Knowledge and Innovation team. And in that role, I co-lead our Blue Carbon program and work a lot on capacity building and knowledge mobilization. I'm joining you today from Vancouver Island, where I live and work on the traditional territories of the Saanich peoples.
Okay, so I think we can flip over to the next slide, Emily, and get started. Perfect. So I'm going to do a quick review of what we covered in our first workshop on ecosystem carbon a couple weeks back. We discussed what carbon is, where it can be found, and why it's so important. We also took a look at a number of platforms and tools that can be used to visualize different pools of carbon across regions. And then we spent the second half of that workshop looking at ways to complete a basic desktop analysis of carbon using ArcGIS, as well as Google Earth Engine, an application that was created. So if you missed out on that workshop and you want all the details, you can expect to see it on our learning library in the next week or so. And if you weren't registered and you want the slides, you can also reach out and we can get those to you. Next slide, please. Okay, so in workshop 1, we covered the differences between green and blue carbon. And we know that carbon can be found across all types of vegetation and sediments, and it's not just limited to terrestrial ecosystems. We know that green carbon commonly refers to terrestrial carbon stores, while blue carbon refers to carbon that's found in marine environments and coastal ecosystems. And similar to how we can find stores of green carbon in peatlands, grasslands, forests, croplands, blue carbon is stored within salt marshes, seagrass meadows, and kelp forests here in Canada. And that image on the right is an illustration of a coastline that has all three of those kind of ecosystems within it. Next slide, please. We know that there is an estimated 327 billion tons, or 327 petagrams of carbon stored in Canada's terrestrial plants and soils. So you can see in that visual on the right, it uses PG as the unit, and those are just petagrams. So 1 petagram is equal to 1 billion tons. Of those 327 petagrams, 94% of carbon is stored in the first meter of soils, and the remaining 6% is stored in plants. And these numbers, if you're interested, they come from a peer-reviewed study completed by McMaster University and WWF Canada. And notably, it does not include carbon found in marine and coastal ecosystems. So these numbers are just for terrestrial ecosystems. Next slide. from that same study by McMaster. So this map came from that study, and it shows a visual of total carbon across the country. So the dark purple indicates larger amounts of carbon being stored. And you can see almost a band stretching from northern Yukon down through northeastern Ontario, and that roughly aligns with where the boreal forest is located, as well as where there is a high probability of peat soils. You can also see over on the west coast, if you take a look, there's a high amount of carbon stored on the coast of the mainland and then on Vancouver Island and north to Haida Gwaii. Next slide. Perfect. So there's currently no comprehensive Canada-wide estimate of blue carbon stocks. However, there are several notable papers with estimates that have been published or are nearing being published through Blue Carbon Canada. This includes a paper on carbon stocks in marine sediments. So the map shown on the right is pulled from that paper, and it shows the percent organic carbon content specifically in subtidal areas. So you can kind of take a look there at the legend and the represented distribution across the country. Other papers that will soon be published include estimates of carbon stocks for seagrass meadows, salt marshes, as well as kelp forests. And Blue Carbon Canada is a great resource for research in this area, so I highly recommend you check out their website for more information. The link will be shared in the chat, and then it's also going to be in the resources slide at the end of this deck. Okay. So that pretty much wraps up what we did in our last workshop. But before we move on to our new content, we wanted to just do a quick poll to engage you guys, see why you may be interested in measuring soil carbon. And so I will just pull that up now. So we'll just take a moment. You should see it pop up on your screen. And you can answer one of the questions, you can answer all of them. You don't need to answer any of them if you don't want to. But this is just to get a sense of who's joining and your interest in soil carbon. And then also at the end, just a little bit of a quiz question to see if you were paying attention. Got some good answers coming in. Just give it another moment for people to get in their answers. I see them coming in. Whoops. Okay. So you should be able to see the results now on your screen. For that second question, it's really interesting looking at the distribution of low priority to high priority items there. It looks like both A as well as C are the most high priority, so feeding into local or regional land use planning, as well as informing future or existing carbon crediting or offset projects, which is pretty neat. And then for the most part, people got that last quiz question correct. There were some incorrect responses, but I'm feeling pretty good about moving on. So let's go to the next slide, Emily. Perfect. And Cal, I'll let you take it from here.
Great, thank you, Claire. It's great to see all those responses. So thank you to all those who participated. I recognize that many people in this call, in this session, come from many diverse set of backgrounds. So given this, I wanted to spend the first 10 to 15 minutes or so providing a bit of a background on soil carbon generally, just to make sure we're all coming from the same page. So for starters, what is soil? it seems like an obvious question. Like we've all touched soil, we've all planted a tree or, taken care of a garden, so we intuitively know what it is. But I wanted to provide here what the Canadian system of soil classification defines soil as, which is in bold there, it says supporting plant growth. So just an interesting definition. And as well, that same group also tried to classify all of soils in Canada. So that's what you're seeing on the map on the right here. The green and the blues would be your forested soils, and the pink would be your organic, your wetland type soils. So you can see that, even in a place so large as Canada, the diversity of soils is quite large, and these are just the great group orders, so they get more specific as well. I thought it's a great map. And secondly, I also wanted to introduce another common word used to describe soils and different surface sediments, which is sediment. And if you look up at the definition of what sediment is, you're going to find often unrelated, even conflicting answers. But typically, sediment's used to describe two things. The first is a particle that's been transported and deposited into a soil, and the other thing or the other definition you might see is it's used to describe soils that lie beneath an aquatic environment. That being said, for the purpose of this webinar, we're going to use the term soils and sediment interchangeably, noting that this might not be the case elsewhere, but just to keep it simplified and make sure we're all talking about the same thing here. Next slide, please, Emily. So when we discuss soil carbon specifically, we're referring to the component of the soils that contain the element carbon. And as Clara mentioned previously, carbon is everywhere. It comes from life, which converts atmospheric carbon into plant tissue. So on the photo on the right here, this is a clear core taken from a peat bog in the Sawtooth region of Northwest Territories in Canada. You can see the living moss layer on the top there grows above the decaying moss layer that's slowly becoming peat, which is a very it's a type of soil that really resists decomposition. And when soil carbon is derived from, living material, we call it organic carbon. It typically has a very high percentage of carbon, usually around 50% by mass. And alternatively, another type of carbon you'll find in soils, which are typically found from the erosion of rocks and different materials. These are called inorganic carbon compounds. And some examples are, your carbonates, your carbides, and your cyanides. But I will note an exception is in coastal ecosystems. A lot of the inorganic carbon is actually derived from living, originates from living organisms, specifically those animals that produce shells. So the shells can get washed up on shore and become part of the sediment as well. Next slide, please. So carbon is very active in an ecosystem, and it's especially active in soils, which is depicted in the schematical simplified version of the soil carbon cycle on the right here. So the flows of carbon between different components of an ecosystem, that's what we refer to as the carbon cycle. When a component stores carbon, it's referred to as either a carbon sink, a carbon pool, or a carbon stock, whereas carbon that's moving between these sinks we call flows, fluxes, and processes. So when carbon moves out of a stock, we call it a carbon source, and when carbon moves in, we call it a carbon sink. And when you take the sum of all these carbon stocks and flows, we refer to that as the net balance of carbon. Next slide, please, Emily. So if we scale this idea up to the entire size of an ecosystem, we can see that more stocks and flows need to be accounted for to try to really understand that ecosystem as a whole. So the schematic on the right here shows what a hypothetical wetland carbon cycle might look like, where you have the atmosphere, the soil, and the groundwater all interacting, and you have these soil processes that are happening from a microbial scale, as well as from a whole ecosystem kind of structure and function type of scale. And then this even scales up to the entire global scale when you look at the impact that this cycling has on the global climate system. Next slide, please, Emily. So from this, we can derive that, scale matters when we want to talk about carbon and soil carbon cycling. And when I say scale matters, I mean that in two different components. The first is temporal scale. So ecosystem carbon cycling will vary over time. For example, an ecosystem that acts as a sink one day might act as a source in the next day. And this can also range over different time scales. For example, you can think of in the summer, you can have an ecosystem that's going to be where photosynthesis is going to be very active and it'll accumulate a lot of carbon versus the winter time where it's less active. You can think on a decadal time scale where you have, your rainy years and your dry years, your cold and your hot years. And you can also think of it as, the ecosystem's response to different actions. This can be, anthropogenic actions like tree planting, or it can also be natural disturbances. So these all act on different timescales. The second scale to consider is that ecosystem carbon varies at different spatial scales. You can think of different stewardship practices here, like taking care of a local garden versus stewarding an entire watershed. And it's just important to note that both of these aspects, the temporal scale and the spatial scale, need to be considered when you're looking at soil carbon cycling. Next slide, please, Emily. So why measure soil carbon stocks? As we mentioned, carbon stocks are components of the carbon cycle that store carbon. Now we're going to transition a little bit more towards measuring carbon, so it helps to be a little bit more precise when we're defining it. So we can define soil carbon stock as the amount of carbon by weight in a square area of an ecosystem to a defined depth. Because typically when you measure soil, you're measuring it to a certain depth in the actual soil. There's two typical units used to express soil carbon. These are kilograms per meter squared and tons per hectare. They both mean the same thing, but are just used to describe different scales. So typically a smaller site will be described in kilograms per meter squared. But if you want to scale up to an entire ecosystem or region, you might see tons per hectare used more frequently. So given this data at hand, how could carbon stocks and measuring carbon stocks support your goals? So remember that we previously mentioned that soil carbon is the largest carbon pool in the biosphere. So measuring carbon can help to identify priority regions around different stewardship. It can help project outcomes related to carbon sequestration. This is especially true over the longer timescales, as plant vegetation typically works on a shorter timescale, whereas soil carbon sequestration works on that decadal to century timescale. And as well, measuring carbon stocks can help inform land use planning and decision making. Next slide, please, Emily. In addition, collecting data on soil carbon may open the door to creating different partnerships, and this can come in the form of research partnerships or funding partnerships, and this can help further support different conservation, monitoring, and stewardship programs. Soil carbon sampling at any level can also help inform baseline values, and these are kind of the foundation for your offsetting projects, your research projects, et cetera, any type of project. And lastly, I'll just mention that the techniques that we're going to go through in this webinar, they're fairly robust and they can be directly used to measure other facets of an ecosystem. Just to name a few, you can use these samples to measure environmental toxins and pollutants. You can look into environmental DNA. You can do a grain size analysis. You could also look at the different isotopes and the radioisotopes in the soils, and you can do some kind of cool dating techniques. Now, these won't be directly covered in this webinar, but just know that the actual methods are the same if you're going to want to look more into those. Next slide, please. So there are many ways to estimate and measure soil carbon. One method is to use existing data on soil carbon and extract those estimates using a desktop analysis. This was covered quite extensively in the first workshop, and that's available online, so I won't go into too many details about that. But below, we just provided a couple resources that they do have these open source kind of carbon maps for soils. WWF-Canada is obviously just Canadian specific, but Soils Grid does the entire globe. And of course, the Canadian Soil System soil database is also Canadian specific as well. Next slide, please. But this isn't a do one or the other type of situation. In most cases, a desktop analysis is a very good first step when you're interested in looking at the carbon in your community or in a region that you're interested in. And then this could then be followed by doing in-field soil sampling. Field measurements can also include various aspects, and you can do them simultaneously. So if you're interested in soils, but you're also interested in trees, these can typically be done in conjunction and together in the same field trip. And if you want to know a little bit more about that, we can, I think Catherine posted the carbon measurement learning library into the group chat, but they have all the different facets there. Next slide, please, Emily. So Why measure carbon in the field if there already exists this data that's available online and it's free and it's open source? The simple answer is that the data isn't perfect. There's large uncertainties in these estimates. And this is especially true for soils. For trees, remote sensing can directly measure the trees because they're above ground and you can get an actual measurement of it. But because the soils are underneath the ground, it's hard unless you have like ground penetrating radar to actually get a remotely sensed idea of that. So it's just for soils, the uncertainty is just a little bit larger. Another thing to consider is that the spatial scale of the estimates is quite large. So I know that, like a strong resolution for this type of work is at the 250 meter square resolution. And some of the project goals might be smaller than that. So you have to sample if you want to get more certainty within that small area. Another thing to consider is that these estimates that are used, these modeled estimates, also rely on conducting field observations. So areas that lack this type of field data also have a little bit of higher uncertainty because they don't have that ground data to work on. Another concern is in regards to data ownership. If you conduct the field work, the data you collect is yours. You have the right to, share it, use it in every way you like. And whereas these open source data are coming from, you're kind of just borrowing that information. And the last thing I'll mention is that any type of carbon sampling, whether you do a single core or you're doing a full-scale project, these can all be used to inform these baseline predictions for creating for building projects out of. Next slide, please, Emily. Okay. So having laid out this foundation for the soil carbon cycle and the introduction to carbon stocks, I want to transition a bit to a more practical sense, focusing on specific methods and protocols for collecting soil samples in the field, specifically for the purpose of measuring carbon stock. I also want everyone to keep in mind that these methods are, as I mentioned before, typically robust enough to incorporate other analyses that you might be interested in. So although we'll be talking about carbon, it's not just carbon that can be done. It's the soil sampling can be done for different analyses as well. So soils vary considerably. That's an understatement, but that's that's just a true fact about soil carbon sampling. And the tools to sample them also vary quite extensively to manage that variation that exists. But no matter where you're sampling, what tool you're using, what soil type you're going to be sampling from, the exact same goal is the same, and that's to collect an undisturbed vertical section of the soil profile. So on the top left here, this is an example of digging a soil pit. And in this, you're exposing all the soil layers, and you're going to grab a sample from each of the different soil layers to get that vertical distribution of the entire soil profile. Another common method is called use an open barrel corer. And this is simply just a tube you push into the ground and you pull it back out. And inside the tube is going to be a nice core. The benefit of that is it's typically quicker and you get an entire full section of the entire soil profile. The cost is that because it's open at the bottom, some of the samples can fall out. And this is especially true if your samples are water saturated. You might not be able to get a sample at all. And this is where closed barrel corers come in handy. And what they mean by closed barrel corers is that when you collect the sample, you're securing the sample in a closed tube so that even if the water, even if the sample is extremely water saturated, it's going to hold it watertight and you're going to reveal the sample and it's going to be perfectly intact. Next slide, please, Emily. I believe, yep, I'll pass this back on to Claire.
Thanks, Cal. Okay, so before we move into the second half of our webinar today, I just wanted to do another quick break. This time it's just going to be one question for you guys, so I'll launch that now. But And so again, totally optional. We're just trying to get a sense if people have been out and taking cores and soil samples before, or maybe this is something that you're hoping to do this coming field season, and this is the first step that you're taking. I'll just give it a few more moments. Okay, I'll end it there. And it looks like there's a decent spread. It looks like the only one that wasn't represented was taking soil or taking sediment samples from a lake, but the majority of folks have never collected any samples before, which is kind of interesting. And then after that, it looks like it's spread between forests, agricultural lands, and then to a bit smaller amount in wetlands. Just kind of interesting. Okay. So, okay. Yep, we'll just move forward a bit. Emily, if you can take us to yep, that slide. Perfect. So we're going to take a look at 3 example scenarios for collecting soil samples. So this will be a large forest, a wetland, and then a coastal wetland. Next slide. Perfect. So each of these 3 scenarios are going to follow the same basic steps shown on this slide. So first, defining your larger study area of interest and its boundaries, then determining how many samples to take. The third step is determining where to take your samples, which is otherwise referred to as developing your sampling strategy. Fourth step is actually collecting your samples as well as any other data while out in the field. And then finally, the last step is analyzing your samples in a lab or sending them to a lab to be analyzed. So we'll review all 5 steps in the first scenario, and then in subsequent scenarios, we'll only be highlighting the steps where there are differences from our first scenario. You can kind of see an illustration there on the right of how you move through these steps and eventually end up with your data. Next slide. So in our first scenario for large forests, we are going to start by defining our study area. This will involve discussion within your team or organization to ensure no key areas are left out. For example, if you are looking at a large forest, maybe including a parcel of the forest where there's been no known harvesting in the past. Alternatively, your study area may also be defined already if it corresponds with a protected area or an existing project boundary that you have. You may also consider this part of the process open to the public by hosting community mapping sessions to determine any boundaries that maybe people are concerned about, or making sure that the boundaries capture key landscape features for folks. And then once you're ready and you've collected all that information, you've had all those conversations, you can map out your boundary using a software like ArcGIS or Google Earth Engine. Next slide. Moving on to step 2, you're ready to determine how many samples to take. So I know we always get questions about, okay, how many samples do I actually need to have? How many can I budget for? And unfortunately, it's not, an easy prescriptive, one size fits all number, unfortunately. And it's important to remember how the number of samples will impact your results. So by sampling a forest or ecosystem, we're selecting a small portion of the total area in order to then approximate or infer the carbon value of the larger area. And this is why it's so important that your samples are representative of the entire study area. Because if they're not, say if they're hyper concentrated in one area that maybe underwent wildfire or floods often, then it won't be actually very representative, and the results that you get won't translate across your broader study area. The number of samples will depend on the following 3 things. So the size of your study area, your project goals, and then your budget. In this scenario, we're imagining a larger forest, so over 10,000 hectares. And I just want to flag that even if it is a larger study area that you have, it doesn't necessarily equal more samples. And an example of that could be if you are looking at a large, uniform, even-aged forest plantation, there will be a lot less variation across the landscape compared to a smaller natural forest that is prone to wildfire. But that being said, the size of your study area is still something that you'll want to consider when you're determining how many samples to take. In terms of your project goals, we want you to really think about the level of accuracy that is acceptable to still meet your goals. So if your goal is to complete a pilot field season and get a general sense of carbon stock that will be used internally by your team only, just for reference, then there may be more room for error when estimating the total carbon stock. Alternatively, if you're intending to report back to funders or maybe set up a carbon credit or offsetting project, you will need a higher level of accuracy, and as a result, you'll need more samples. And then finally, there's budget. So we can go to the next slide to talk a bit more about budget. Okay, so budget will likely be the main factor when determining the number of samples you'll collect. When planning your budget, it's really important to consider staff time, equipment costs, transportation, shipping, as well as lab fees. And in some cases, if you are traveling to remote areas, you may need to factor in accommodations and food as well. I've included an example of lab fees for analyzing soil samples for carbon only. And so you can see there, it's set at $105 Canadian per sample, and that's for 10 samples. And you'll be able to obtain a quote from the lab that you're working with ahead of time, and you can even ask for several quotes. That's totally normal, especially if you're unsure of how many samples you're going to end up with. You can say, okay, give me a quote for 10 samples, 50 samples, and 75 samples. And this is important because the cost per sample does go down to a certain extent with more samples that you do. It's also important to confirm if the pricing that they send you includes disposal fees, or if your team will manage that, because again, it'll impact your cost per sample. And if you maybe want to save some money, you can choose to dispose of the samples yourself. So working within the constraints of a budget will mean that you need to ensure that your priority areas are captured by samples. And so this could mean stratifying your study area into unique units. And we'll speak more about this on the next slide. And so the last thing I'll mention about budget is that you can also complete your sampling in phases over several years to spread out costs. So after that first pilot year, when you've got a little bit of data on your study area, your site locations could actually be further refined depending on those results and feedback from your field crew. Okay, next slide. Perfect. So in the next step, we will be determining where to take our samples by deciding which sampling strategy works best for you and your team. So there are 4 main sampling strategies you can use, and each has its own pros and cons depending on your goals, study area, as well as budget. So we'll just go through each of these, and you can see that 3 of them, the first 3, also have illustrations on the right-hand side that correspond. So for random sampling, you set up a grid or a transect with numbered intercepts, so points where your grid kind of intercepts with itself. And then you can use a program to randomly select which intercepts you will be using to set up your plots. This sampling strategy is used when your study area is uniform or in cases where you don't know that much about the ecosystem. And it's generally used as the default strategy. So if you're not sure, what to do, I would highly suggest using random sampling strategy to start and go from there. For systematic sampling, you evenly distribute your samples across the study area. So this is really good if you have a smaller study area that is more uniform. However, you must have a good understanding of the variability across the entire study area. And the reason for that is you don't want to accidentally assign a bunch of plots where maybe you're overrepresenting a certain feature on the landscape, and then you're not capturing another feature. And so if your team has a really good understanding of the landscape you're working in, and maybe it's a bit smaller, this might be the sampling strategy for you. Then for stratified random sampling, you first divide your study area into strata or smaller units based on the landscape features. And then from there, you randomly select plots within those units. This is really good for larger study areas where there is a known variability, so it's not uniform, and you want to make sure that that variability is captured when you're doing your sampling. And then the last point is just convenience sampling. So this is exactly what it sounds like. It's basically where you set up plots based on what is easiest for you and your field crew. So what is the easiest spots for you to access, or the most efficient places to get from your home base. Okay, next slide. Perfect. So for more information on sampling strategy, you can refer to the guides in our carbon measurement learning library. In the next month or so, we'll also be sharing a guide specific to sampling design, so you can look out for that. In order to map your plot locations, you can use a number of tools, including Google Earth Engine, ArcGIS, QGIS, as well as manual sampling selection. And if you're new to any of those programs and you'd like assistance mapping out your plot locations, feel free to reach out to our team and we can assist you no matter kind of what phase you are at that process. Next slide. Okay. After determining your sampling strategy and your plot locations, you're now ready to head out into the field to collect your samples and your site data. We have two guiding documents you can refer to for the exact step-by-step methods when sampling in peat and non-peat soils. These can be found again in our learning library, and they also include all of the equipment that you could possibly need for sampling soils. Other protocols do exist, like those by Bansal et al., as well as the National Forest Inventory and the BC Assembly of First Nations Carbon Toolkit, so feel free to check those out. And like Cal mentioned just before the poll that we did, there's a couple different ways that you can go about sampling in forested areas. So most likely the tools that you would be using would be a closed barrel core or setting up soil pits. And soil pits typically take longer amount of time. So if you have a lot of sampling to do, you probably won't be doing soil pits at every single plot. Maybe you'll do one just to test it out and build out that skill set, but likely you'll be doing closed barrel coring in the forest just because it can be a lot quicker. Okay, next slide, please. And here you can see just a snapshot of our guide on the left on carbon measurement in non-peat soils. That's kind of what it looks like in the guides. We also have quick videos for each guide, so that way you can watch, a 5-minute video instead of reading a huge PDF document, just to get a sense of what you're in for and what the methodology involves. We also, on our learning library, have data sheets that you can download and then you can use as is, or you can tweak to suit your needs. Okay, next slide. Okay, I'm going to hand it over to Cal.
Great, thank you, Claire. So I'm going to be discussing briefly just how to measure soil carbon specifically in wetlands. So I'm not going to cover steps 1 to 3 that Claire went over in a little more detail because they're pretty much the exact same, where you define your study area boundary, you determine the number of samples, and you determine where to take those samples from. And you can use the exact same protocols that Claire mentioned and that are going to be a part of the protocol guide. One thing I do want to mention for wetlands, and this applies to coastal wetlands, are that typically wetland soils tend to be deeper, and because of that, they're more costly per core, just because you have to analyze more samples and each sample has an associated cost with it. And because of this, a lot of researchers have tried to really determine what's the most cost-effective strategy for sampling in these wetland environments. I've attached one paper that I really like. So this was by Young et al. It was a paper out of Australia looking at, what's the minimum amount of studies or a minimum amount of cores I need to take in an area, and what's the minimum amount of samples I need to take per core to really accomplish different project goals and different project outcomes. So here they defined having 80% statistical power as their project outcome, and they determine how many samples they need based on that. So that's a good paper to look forward to. I won't go into too many details, but that's just one little wetland tweak that I would make that's different than forest. If you can go to the next slide, please, Emily. So when you're collecting wetland soils, the step 4 is going to be different, and that is, the actual collection of the sample. And there are a couple of considerations that need to be made specifically in wetlands. The first is that the depth of the wetland soil might be potentially much deeper compared to a forested area not too far away. So this core on the right here, this is a peat core I collected in the Mackenzie River Delta. This is in the Sawtooth region of the Northwest Territories in Canada. And this core was collected 2 meters into the peat, into the bog. So it was a 2-meter core. So yeah, the depth really has to be accounted for. And if you're sampling every 10 centimeters, that's 20 samples for that just single one core. So that's, peat depth in this case, but soil depth in wetlands generally has to be considered for. The other thing to consider is where the water level is at. So if the water level is at ground or above ground, then all of the soil is going to be saturated, and you need a closed barrel corer to really secure that sample so the water doesn't rush and flood all the soil out of the barrel. However, sometimes you'll be in a wetland, or maybe it's the dry season, or maybe it's in the fall where the water table drops to maybe 30 to 50 centimeters below the ground level. You might have to use an open barrel core like Claire mentioned, just in that top little bit till you hit the water table, and then you have to switch instruments and go to this closed barrel core. So that's where you have to really look at what soils are water saturated and which soils are not. And the last thing you have to consider is the type of soil. So in this scenario on the right, this was very peaty soil. It was very water saturated. The cost of it being water saturated and peaty was that it was very loose. The benefit is that it's really easy to just collect a clean core because the barrel can cut right through the peat. However, if you were to try to do this in a more like grassy type environment where there's like a very complex root network, the roots are going to get away in the way of the core, and you're going to need a more appropriate tool. So in that scenario, you want to use like a Warden or a box core in that situation. Now, I'm not going to go into too many I'm not going to go cover every single tool that could possibly be used in every environment, but luckily there is an existing paper that does, and it covers pretty much extensively every wetland environment you could encounter in Canada and the tools to approach that. So yeah, if you look at the Banz-Lytel paper, it's really a great resource to have. Next slide, Emily, please. So as part of our tools in resources library that Claire had mentioned previously, we do pretty extensively cover these wetland and peaty soils, and we specifically cover how to, collect a high quality peat core using the tool is often called a Russian peat core or a Macaulay core. And this guide is great for outlining the different types of equipment, what environments are going to be appropriate for. Site selection is also covered in this guide, as well as packaging the core and scaling up the findings. So it's a great resource, so please find those in the learning library. Next slide, please, Emily. And this is just an example of what it might look like. And you can also find videos that show you how to do this online as well. But this is just to set you up using that Russian peat core in the wetland environment. But yeah, before you do any wetland sampling, sometimes it's a trial and error type of situation, but really consider those 3 factors: the depth, the water saturation of the water table, and the type of soil that you're going to be looking into. Next slide, please, Emily. I believe, yeah, we'll be transitioning to a different wet lab type, and I'll pass the mic to Marianne.
Thanks, Cal. I'm just going to pop in here for a few slides to talk about some of the considerations that are specific to coastal wetlands. As Claire and Caleb said, there's a lot of overlap in terms of methods among different ecosystems. So really steps 1 to 2 are the same in terms of defining the study area boundary, determining the number of samples. It's going to come down to budget and your project goals. But just a few things, a consideration specific to coastal wetlands like salt marshes and seagrass meadows in terms of the other 3 steps. So for where to sample, there's likely to be differences in physical and biological characteristics of the system that you may want to consider. So a fairly obvious one for marsh is in terms of upper and lower marsh, which may have differences in terms of elevation, in salinity, and in terms of plant communities. So that might be one approach to dividing your study area before you start sampling, is to stratify that into upper and lower marsh. Likewise with seagrass, the similar approach might be done if you have both intertidal and subtidal seagrass. You may want to make that division and then decide on your sampling protocols. Another approach for salt marshes is to take a systematic approach and work along the elevation profile. So taking samples along a transect that runs perpendicular to the sea. So going from the sea towards the back of the marsh at different elevations. There's no right or wrong way to do that. There's just some of the considerations you might want to think about for those types of wetlands. Also, there's logistical considerations around access. You're likely, particularly in the intertidal areas, need to consider whether you can access those sites by land or whether you're going to need a boat. And in terms of getting the equipment there and then going on to processing the samples afterwards, that's an important consideration. Also, timing of taking the samples and fitting those in within tides. The right tide windows. And then for seagrass meadows, if you've got mostly subtidal seagrass, then you may need to consider using dive or snorkel or scuba to collect those samples with it, which is a challenge all of its own, but it's definitely doable. Next slide, please, Emily. So in terms of collecting samples, similarly to what Cal was just saying, sediments are likely to be very wet. You need to consider the tides, but the sediments are going to be wet anyway. So one of the key things is making sure once you've collected the samples that you keep those cores upright, so you're not mixing the sediments. And that's particularly important if you want to look at the dating, the date of the sediment profile. So how old the sediment is in different layers of that sediment profile. The other thing to consider in terms of the actual analysis is, particularly for seagrass sediments, you're likely to have quite a lot of shell fragments in that, or shell hash, which is going to increase the amount of inorganic carbon. So when you're considering your lab analyses, you want to factor in measuring both the organic carbon and the inorganic carbon, so you've got an understanding of how much of that is how much the shells are contributing to your overall or your total carbon. And then in terms of methods, for marshes, there's a lot of overlap with like peat wetlands. Again, there is no one size fits all, and it's down to budget and what works best for your team. You can use a lot of similar methods. One method that was actually developed specifically for seagrass is using a PVC pipe, and it's a very low budget and quite simple to use. So you have a 3 or 4 inch PVC pipe that's pushed down into the sediment. And then to extract that, you dig around the bottom, you cap it at the top and then cap it at the bottom and then bring that out. And you can do that with dive teams. So that's a useful way for sampling within seagrass meadows. Next slide, please, Emily. And this is, sorry, go back one. Thanks. This is not specific to coastal wetlands, but going back to what we said earlier about doing different types of analyses, we found benefits with collaborating across teams to take samples from the same area, particularly where it involves dive teams or snorkel, which can get quite labor intensive and quite expensive quickly. So recently we went out with a team of First Nation government and NGO staff. We all had different interests, but the same, we kind of collaborated together to collect the samples. And that just reduced the costs in terms of labor, but also in terms of the shipping costs to get samples out to the lab and sharing equipment. So for those samples, we were sending them off to look at both carbon and heavy metals and radioisotopes. And going back to what Claire again said, reaching out to university labs can be really helpful in terms of figuring out those different analyses. And they do approach labs and ask what they're able to do. So they may advertise the services of doing one particular type of analysis, but actually be able to do maybe carbon on top of that. So it's worth reaching out and asking those questions. Okay, thank you. And I think I'm passing it back to Claire.
I think Cal, you were gonna tackle this part. Yeah.
No, thank you, Marianne. I love hearing about the coastal environment. Honestly, I gotta be honest, the photos from the coastal wetlands are just so beautiful. I love them. So thank you for that. So I'm gonna just discuss a little bit about the next steps. So whether you're sampling in a forest or an inland wetland or a coastal wetland, the same analysis will be run for your samples, and the lab will likely ask for similar requirements for each sample. One note, it's very important to package your samples in accordance to the lab instructions. As Marianne said, like different labs, whether it's private, government, or university, they might have different requirements for how they want that. So make sure to really follow those instructions specific to the lab you're working with. And then lastly, I want to discuss a little bit about taking those individual results and scaling them up to the study area to provide a whole ecosystem or a whole study area type of estimate. So next slide, please, Emily. So to begin, I just wanted to show a quick example of what processing a core in a lab might look like. So this is a core taken by me and a couple of my research partners from it's a freshwater marsh in southern Ontario, and there was a large root system, so we couldn't use that P core, but we used a Wardener box core, and it grabbed this big square sample. Also, if you're thinking, is the sample inside of like a rain gutter, then the answer is yes. That's, as you know, we're talking a lot about low-cost solutions to sampling. So if something already exists, you don't have to pay a lot of money to find a custom mold. This is one of a great low-cost solutions to just protecting that sample in that plastic frame that's already built, and you can go to your local, home hardware just to pick up. And then on the right, we have some of my colleagues that are dividing up the sample into individual sections, and then those will be ground and analyzed further. If you go to the next slide, please, Emily. So the photo on the right here, this is what the samples will look like after what's called a loss on ignition analysis. So starting from the bottom right and moving up to the bottom, or moving to the bottom left in like a zigzag fashion, you're going from the top of the core to the bottom of the core. And by burning the samples, you can get a very accurate estimate for the organic carbon in a soil sample. And so on the bottom right here, you had very high organic carbon. And then you're moving into that clay material in the bottom left, and that would be much less organic matter and therefore much less organic carbon. Another example I wanted to show is that Marianne mentioned a bit about dating and different sub-analyses you can do on your core. So on the top left here, this is a core from the Lake Simcoe region in southern Ontario that I took a couple years back. And that herbaceous peat was a mix of grasses just at the bottom of the core. So it was around 1.5 meters below the soil surface. And after radiocarbon dating, I found out that grass is actually 11,000 years old. So pretty much right after the glaciers left southern Ontario, this river would have been infilled with different grasses, and they're still there today, and just lying underneath the ground 1.5 meters, which I thought was cool. And then in the middle of that core, there was this big piece of wood that I found, and I also radiocarbon dated it. So it was 50 centimeters below the surface, so about a meter higher up than that herbaceous peat, and it was dated to around 4,000 to 5,000 years old, and it looked pretty much as it did back in that time. So it just really shows how great these wetlands are in terms of slowing down decomposition and preserving that carbon for a long period of time. Yeah, just want to show a little bit about what the lab process might look like, because oftentimes you'll send the samples and you'll just get back a data sheet, but this is what they're actually doing. Next slide, please, Emily. So in terms of, next steps, like you said, you have the samples collected, analyzed in a lab, and now you have your data. And the lab might not necessarily give you that soil carbon stock that you're looking for, so you might have to calculate it yourself. It's relatively simple, and it just requires a minimum of two analyses. The first is bulk density, and bulk density is simply a measure of how much soil there is in a sample, and you find it by measuring the weight of your sample divided by the total sample volume. This is a foundational measurement for all analyses, whether you're doing environmental toxicology, whether you're doing carbon, or maybe you're doing nutrient cycling and you're looking at the nitrogen and phosphorus. Bulk density is kind of the foundational measurement for all of these. And then secondly, you're going to want to also look at the carbon content. As previously mentioned, this could either be inorganic carbon content, or both, and typically called total carbon content. But it's the measure of the relative amount of carbon in your samples expressed as a percentage or a decimal form. Now, for each soil core, the carbon stock is simply measured by just multiplying these two measurements, and then you're going to also multiply that by the soil depth, because the more into the ground you go, the more of that soil profile you're capturing. And that's a very important measure, because you're going to get different measurements if you only study the top 10 centimeters versus if you do the full, 30 centimeter or a meter depth. So this is how you calculate the carbon stock of one single core. But you also might be interested in, okay, well, what's the carbon stock of the entire study area that I'm interested in? Emily, if you don't mind going to the next slide. Scaling up from single sites to entire study areas can be done typically using one of three methods. The first I'll describe here is called direct field-based estimation. In direct field-based estimation, the sites are averaged within an area and used together to approximate a larger area. Therefore, the amount of uncertainty is directly related to the amount of samples you collect in that area. The other types of analyses are mass balance modeling. For those familiar with the Canadian Forest Service carbon budget model, that's an example of a mass balance model, as well as there's remote sensing methods that use either satellite, airplane, or drone-based remote sensing techniques, and they combine that with field observations to kind of produce a value of carbon in a certain area as well. Now, for each of these different types of scaling methods, the goal is to approximate what's called the aerial carbon stock, and this is sometimes referred to as the average carbon stock, but they both mean how much carbon is there per square area of land, almost like from a bird's eye view. That's what it's trying to capture. And as mentioned previously, this can be expressed in kilograms per meter squared, which is what you would typically have at a smaller scale. But some researchers or some projects are so large that it just makes sense to use tons per hectare, because you're working on the scale of hectares, not square meters. And then if you define that depth, you might also see carbon density used, and that's simply just the same metric as aerial carbon stock, except you're defining the depth that you actually sampled. So it might be 30 centimeters, 50 centimeters, etc. And then on the right here, this is just a little schematical representation of how that direct field-based estimation would work, where you're just in a certain area, you're going to measure the aerial or average carbon stock. And then as you scale up, you're going to find the average across the different regions, and then that's going to inform the carbon stock as you scale from that small site all the way up to the region. The last thing I'll mention on this slide is that you also might be interested in knowing what the total carbon stock is. So as Claire mentioned previously, when we look at those Canada maps and we're looking at those petagrams of carbon, those billions of tons of carbon, those are obtained by multiplying the aerial carbon stock by the total area of the forest or wetland or coastal habitat that you're interested in. So it's a simple calculation just to get that as well. Again, typically represented in kilograms or tons. Next slide, please, Emily.
Okay, I can take over,
Oh,
Cal.
thanks, Claire.
Yeah. Yeah, thanks for going through each of those three scenarios for us. And so I'm just going to give a quick summary of what we've discussed today. And then if you guys want to start thinking about any questions that you have, or if there's certain slides that we should go back to, that'd be great because we're going to come up on the Q&A period after this. So, to summarize, today we discussed how taking soil and sediment samples can provide you with a baseline estimate of carbon stored within your study area. And we also chatted a bit about how before you actually head out to collect your samples, get out into the field, it's really critical to have a clear idea of the available budget, your staff capacity, as well as the objectives for your project so that you could develop the best sampling strategy. Especially knowing, how much room for error you have with accuracy, that will tell you kind of how many samples you need to collect. And then also that this data, after you've collected it, it's been processed in the lab and you've gotten it back, you can use it to guide environmental monitoring programs or support land use planning initiatives. And with that in mind, be sure to discuss your sampling strategy across teams or departments to see if you want to include any analyses beyond carbon. And include that, maybe not for all of your samples, but for specific samples in different areas, as well as discussing how to best share results within your organization, your community, or perhaps beyond. In some cases, if you are looking to share your data and your results beyond your immediate community, maybe with academic partners or NGOs, you may also want to consider building out a data sharing agreement with external partners. And if that's something that you have questions about, you can come to us as well if you just want to talk about, what that could look like. Okay, next slide. Okay, so this slide, as well as the next slide, which we can flip to, are just a list of resources. So that first one was, research papers, websites of interest. And then this one is a breakdown of possible labs that you could reach out to. It's non-exhaustive. So if you want, you can easily Google, labs and then your location to find one that's near you. There's some private ones listed here and linked, as well as some government labs. And then for university labs, as well as college labs, we highly recommend reaching out to them directly to see if they offer services for soil analysis to the public. In many cases, this might be possible. However, they don't typically, advertise or promote that on their website. It's done kind of behind the scenes. So you need to reach out to understand, what their rates may be or if they only do it at certain times of year. But that's also an option. Okay, so we can go to the next slide. And I think before Q&A, actually, Cal had a couple kind of recap questions just to test out if you guys were listening. So I can launch that now. Doo-doo-doo. And again, you don't need to take part in this if you don't like, or you can answer one, or you can just go ahead and see what you want to answer. But yeah, there's two basic scenarios there for you. We're getting some answers in. I'll leave it open for a few more moments. And then, yep, at the same time, if you guys have questions, feel free to pop them in the Q&A now, or you can also hold them until, yeah, we're ready. Okay, I think I'll end the poll and then we can share results back with the group. And Cal, can you see the results on your side? You might need to click that. Okay.
Yep, they popped up. Thanks, Claire.
Okay. Amazing.
So for the first question, I'll preface this by saying there's no right answer here. As you know, Mary and Claire and I have been discussing. It's just about collecting that high quality sample. This was kind of so there's 3 general answers here: the open barrel corer, the rotary auger, and the soil probe. Now these things also, much like this field, they mean different things in different scenarios. But the open barrel corer is the one that's going to guarantee that undisturbed full soil profile. The rotary auger has a tendency to break up the soil a little bit more, and you can't get a strong bulk density estimate. I have seen people pair rotary auger samples with these bulk density discs, and that's a fine solution of itself. Yeah, that's good for question 1.
And in question 1, you could use a soil probe, but it wouldn't be to actually collect the soil. It would be initially to kind of get a sense of soil depth.
Yeah, the soil probes
It's
are
like
great for surveying. And some soil probes are cool, they'll actually collect a little sample, but it has the same issue with the rotary auger where maintaining the integrity of the core is quite difficult, so you don't get as precise of a bulk density estimate with it. But you can get your organic carbons and that stuff as well.
And then Cal, what about question 2? It looks like people were swayed towards 1.
Yeah. No, I think I talked about it a little bit when you have, the water table might be above ground or below ground, and sometimes you might have to be a little bit creative. If your closed barrel core isn't cutting through the dry material, you might have to use that open barrel core until you hit the water table. But at that point, it might be too water saturated, and the open barrel core might not hold it, so you have to switch equipment. I get teased a bit because I bring a bread knife into the field, because sometimes when it's fibrocky, you gotta, there's no right strategy. But yeah, C was definitely the answer I was looking for. Thanks, Claire.
Okay, Emily, I think we can move to the next slide. Okay. I haven't seen any come through the Q&A, but maybe I'm missing something.
Yep, there's actually one that has come through. Just came in a couple minutes ago from Maxine, who asked, how can we avoid overestimating carbon stocks in forest when there are large rocks in the soil? So that's a good question. I don't know, Cal, if you want to take that one, how we can avoid overestimating?
It's a great question. One clarification is, are the rocks above the soil? Like, are they big boulders? Or sometimes you can also find rocks beneath the soil. So like you go to take a soil sample and you're expecting to go down 30 centimeters, but you hit a big boulder at 5 centimeters in depth. The best way to account for it is to do a survey using a soil probe and measure the different soil depths. And that will actually give you an estimate of what the average soil depth is in the study area you're looking for. So it accounts for all those locations where the soil might only be 5 centimeters because there's a big rock underneath, but it'll also capture the different areas where there might not be rocks, and you can get a full, 30, 50 centimeter sample.
Great. Okay, thanks, Cal. I think that answers the question. Maxime, if you have any other follow-up questions or anything, please feel free to add them to the Q&A. And for others as well, if you have any questions, please do add them to the Q&A section. It's just at the top in case folks can't find it, or might be actually at the bottom for you. You can just scroll over and find mine's under the More button, and you can type in questions under the Q&A there. So we'll give it another minute just to see if any other questions come in. But if not, we'll move along. Thank you for kicking that off, Maxine.
Emily, did you maybe want to share a bit about the community of practice?
Sure, yeah, I can do that. Well, folks are thinking of questions for you. So I wanted to take a moment to share with everyone that we do have a carbon measurement community of practice. That is, we have two. We have one that's focused on blue carbon that Marianne leads, and then we have one that Claire and myself lead on the terrestrial side of things. So we meet quarterly, just virtually, and we share knowledge and exchange ideas and tackle challenges that we have with carbon monitoring. So if that's something that's of interest to you and you'd like to join, please do let me know. You can send us an email again at that science@WWF-Canada email address, and we'd love to have you join. We are also going to be meeting in person this fall, so that's another exciting thing to look forward to. I see that Chris has raised her hand, but I'm not sure if, Chris, you're able to come off mute and say something. Or you might have to put it into the chat. I'm not sure how to grant access. Actually, here, Chris, you should be able to come off mute now if you wanted to say something. Oh, no problem. It was a mistake. Okay. Okay, well, seeing as no other questions, I guess we'll move on. But again, as we're wrapping up, please feel free to ask any other questions that you might have.
It looks like we just got a question about sampling in New Brunswick. So last year we did actually do some training out in New Brunswick, some in-person training out there with a number of different groups. And so if you are based in New Brunswick and you're interested in, connecting with us, again, just reach out and maybe we can coordinate some additional training, or maybe even just like knowledge exchange between those folks that we trained last year out in that area. And yeah, so this is just a reminder, if you haven't registered for the next few webinars, we've got 2 more in this series coming up. So one on March 5th for vegetation and trees. And then on March 26th, we'll be talking about drones and ROVs and how they can be used for environmental monitoring. And I believe a link or 2 links were just added in the chat, so you can click on those to register now. And next slide. I think that's it. Yep. So there's our email address again. So if you do have any questions after this, you can reach out. But yeah, thank you everyone for joining.
Yep, thanks everybody.